వాళ్ళకు నేను ముందు చెప్పేది ఏంటంటే
Okay, good morning all, good morning. Okay, so today we will understand the basics of the cell signaling topic. Okay, so friends, these are all the details about the upcoming batch, that is the for CUCET Embark batch, which was started from 9th February, that is from yesterday. So in this the people whomever preparing for the CUCET, so the entire syllabus will be covered in this particular batch. And friends, as you know that IIT-JAM exam is going to be conducted on the 13th February 2022. So immediately after the exam, we are conducting a session on paper analysis and discussion, where you can understand the cutoff, insight of the question paper, difficulty level of the question paper. So please kindly do not miss this particular class, 13th February 12:30 p.m. And next, another important point here, there is a very limited offer is going on. If you take a 12-month subscription, there is a free of 3-month subscription. If you take 24-month subscription, there is a free of 4 months subscription. So please kindly take this offer. In order to get 10% of discount, please kindly use my code ChanduLive. Okay, so friends, let's continue the important topic, cell signaling. So friends, in this particular cell signaling topic, very surely we can expect the questions. We can expect the questions in the 1 marker also. Even friends, we can expect the question in terms of 2 markers also. Okay, so now what is cell signaling? Why the cell signaling is most important actually? That's the question. Cell signaling is most important because friends, every function performed by the cell, signaling is utmost important, as we know very well. You take an example, if the cell wants to survive, if it want to perform any functions, even if it want to divide, differentiate, and friends, even cell wants to die, that is the apoptosis or the programmed cell death. For every single process, the cell signaling is very important. And what are the type of cell signaling? There are many type of cell signalings are there. These are all the forms of major cell signaling, that is endocrine, paracrine signaling, autocrine signaling, then friends, contact dependent signaling. You can see this picture clearly. What do you understand by the word contact dependent signaling? Friends, contact dependent signaling is a process. All of you listen. Contact dependent signaling is a process where the signal releasing cell and the signal accepting cell both are very close proximity to each other. Do not forget that particular point. Means this is the cell which is releasing the signal, an example, and this is the cell which is accepting the signal. So friends, the signal releasing cell and the signal accepting cell both has to be associated very closely to each other. That kind of signaling only we can call contact dependent signaling. Don't forget the statement. Next one friends, autocrine signaling. This is very important. What do you mean by the word autocrine signaling? See here, this is the signal releasing cell. It has released a particular signal, and what is the point you can understand? That particular signal will come and associated with the receptor present on the same cell. That is what exactly, friends, we will be calling it as autocrine signaling. What is the best example for the autocrine signaling? You can take cancer cell. Is it? Friends, what is the speciality of a cancer cell? As you all know that cancer cell will undergo the rapid growth or the continuous growth. As you all know that whenever in order to grow, cancer cell require the growth factors. Without growth factors, the growth cannot be rapid. So to grow rapidly, autocrine cell continuously starts releasing the signal. Now that particular signal will come and associated with the receptor present on the same cell. This is a kind of autocrine signaling. Don't forget it. Right. Next kind of signaling is what, friends? Paracrine signaling. What is paracrine signaling? You can see here. This is the signal releasing cell. This is the signal accepting cell. This particular signal releasing cell associated with the signal accepting cell with the help of an intermediate signal molecules. I mean to say, you can use a word carrier molecule. So listen, this is the cell released a signal. This particular signal with the help of a carrier molecule or with the help of a intermediate molecule, friends, it will go and reach the targeted cell. This is what exactly, friends, we will be calling it as paracrine signaling. Please kindly don't forget this. Next one, what we can call it as? We call it as endocrine signaling. What is this particular endocrine signaling, friends? All of you listen to me carefully. This is the signal releasing cell and this is the signal accepting cell. You know, right? Once the signal released from the signal releasing cell, that particular signal travel via blood as their stream of transport. How, friends? How they are traveling? They are traveling via blood as their stream of transport and reaches the targeted cell. So you can ask me what kind of signal molecules basically uses the endocrine signaling. You all commonly know that best example that you can take is hormones. Hormones are the one which can show this particular endocrine signaling, where the signal releasing and the signal accepting cells are entirely different. Please kindly don't forget this. So friends, these are all what? Types of cell signaling. Simple point. Now here comes to a question. We understand there are different type of signal molecules are there. Now friends, whatever the signal, ligand, primary messenger, whatever we may call, if they want to show their activity, where they have to associate. All of you keep it in your mind, friends. If they want to show their activity, they have to associate it with the receptor compulsory. Do not forget that particular point. They have to compulsory associated with the receptor. Without associating with the receptor, they cannot show their action at all. Now friends, let's try to understand what are the types of receptors. There are many major classes of cell surface receptors are there. What do we call it as, friends? Cell surface receptor and ion channel, G protein, enzyme linked. Actually, these are all the examples for what, friends? These are all the examples for the cell surface receptors only. One we called ion channel, one we call G protein, another one we call, friends, enzyme linked receptor. Please kindly don't forget the statement. See, these are all the example of cell surface only. Why you call it as a cell surface? First of all, you are calling it as a cell surface. They are present on the membrane. Now here comes a question. What is the question? Based on what criteria we categorized the receptors? Based on what we categorized the receptor? Friends, all of you listen. Based on the type of ligand. That is important statement you have to remember. Based on what, friends? Based on the type of ligand, means based on the signal molecule. See, you know, right? There are two type of signal molecules are there. One we call it, friends, hydrophilic signal molecule. Another one, friends, we will be calling it as hydrophobic signal molecule. Correct na? One is hydrophilic, another one is what, friends? Hydrophobic signal molecule. All of you keep that point in your mind. So when you understand hydrophobic signal molecule, what is the statement you have to remember? Hydrophilic means, friends, what? All are water soluble. Correct na? So you know very well, water soluble means they cannot cross the membrane. So friends, undoubtedly all the hydrophilic signaling molecule starts using cell surface receptors only. Do not forget the statement. Next one, hydrophobic signaling molecule. So they can easily cross the membrane, is it? Of course, yes. So friends, always keep it in your mind, they cannot use the cell surface receptor. Since they are crossing the cell, friends, they are going to use the intracellular receptor. Please kindly do not forget this particular statement. Now, what are the type of receptors? I said type of cell receptor, ion channel linked, G protein and enzyme linked. So let's try to understand one by one first briefly. Okay, ion channel linked receptor. What's the point you have to understand? Ion channel linked receptors means what, friends? Remember everyone, these ion channel linked receptors are closed every time. They never ever opened actually. They will be closed every single time. Friends, they will be opened only whenever a ligand will come and attaches. What is ligand? It's a signal molecule. You can also call it as primary messenger. Anything we can say. Now the statement comes here is very clear. When they are getting opened, that is the point. So whenever a ligand comes and attaches, what is this function of a ligand? This is the ligand which is actually involved in the movement of ions across the plasma membrane. See friends, if this particular ions wants to enter inside, what they required, this particular receptors to get opened. So for that purpose, what you have to understand, whenever a particular ligand comes and attaches, whatever the ion channels closed so far, they will get opened. Now the ion starts entering inside. Very simple. Now anyway, we are going to read the G protein coupled receptor in the next slide very briefly and elaboratively. So before that, let me explain you what is enzyme linked receptor and what kind of ligands will use this. Friends, this is more important for the exam. What is that? What kind of ligands or what kind of signals uses the receptor? If you take an example in the next slide, I will explain you most of the hormones and most of the neurotransmitters, most of the hormones and most of the neurotransmitters will start using this particular G protein linked receptor. Kindly do not forget it. But friends, when you look into the enzyme linked receptor, what you have to understand, they can be majorly used by the growth factors. That's a very important point, friends. Almost all the growth factors present in the body, they start using this particular GPCR only. What is the best example you can take? You can take epidermal growth factor, you can take insulin growth factor IGF, you can take PDGF, platelet derived growth factor, you can take an example of NGF, nerve growth factor, EGF, IGF, PDGF, NGF, and many, friends, many. You can take another example of FGF also, fibroblast growth factor. These are all the type of growth factors start utilizing this particular enzyme linked receptors in order to show their action. Now the point comes here, how does these particular enzyme linked receptors are getting activated and how they are going to show their action actually. All of you listen, friends, everyone listen to me. Every enzyme linked receptors are inactive monomers. There are two monomers are there, they always associated like this. That's why we can call them inactive monomers. Now see here, they are not associated with each other, they will be like this, friends. Now all of you has to understand what are the various kinds of domains that are present in this particular what we can say enzyme linked receptor. Friends, remembering the domains is extremely important for the exam. It contains an extracellular domain. Friends, sometimes instead of using a word extracellular domain, they can also use a word ligand binding domain. Anything is same. It contains one extracellular domain, and friends, it contains one transmembrane domain. What do you mean by the word transmembrane domain? Through which it will transpan the membrane. And what else it contain? It contain very important one, friends. We can call it as a cytosolic domain. What domain we call it as? A cytosolic domain. All of you please keep it in your mind, friends, without fail. The cytosolic domain having intrinsic kinase activity. What kind of activity it contain? It contain intrinsic kinase activity, which is having autophosphorylation activity. Means they can phosphorylate on their own. But the point is very clear. When they phosphorylate, will they phosphorylate every time? No, that's not possible. They are going to phosphorylate whenever ligand comes and attaches. If ligand did not come and attaches, they never undergo what we can say that they never undergo the process of autophosphorylation. Don't forget that. Okay, now let's try to understand the most important for the exam, that is the GPCR. So most of the people never understand what exactly the word G representing. So friends, G basically representing guanine coupled. What, friends? Guanine coupled. And I want you people to remember, friends, when compared to the other cell surface receptor, all of you keep it in your mind, GPCR or G protein coupled receptors are the largest and most diverse group of membrane receptors in the eukaryotic organism. Please kindly don't forget the point. And friends, all of you keep it in your mind, the general functions of our body. Say an example I am talking vision. Right, vision, hearing, touch, hormonal action, all these major processes can be mediated by the GPCR in our body. So same. Now let's try to understand the clear structure of the GPCR protein. Now all of you listen. This GPCR is a 7 transmembrane protein. What, friends, it is? It's a serpentine protein, which means which can transpan the plasma membrane 7 times. And while discussing the enzyme linked receptor, I said that there are different type of domains are there. Same way, friends, even in the GPCR also there are different type of domains are there. One we called as extracellular domain, one we called transmembrane domain, another one we called, friends, cytosolic domain. Same. And if you look into the transmembrane domain, it is not transpanning the membrane just one time. It will transpan 7 times actually. That's why we can call, friends, 7 transmembrane. What do you call, friends? You call it as a 7 transmembrane receptor, or friends, we will be calling them as a serpentine receptor. 7 transmembrane as well as the serpentine receptor. Now friends, listen. When I use a word, they are transpanning the plasma membrane. There is no doubt about it. But how? How a cell surface receptor can transpan the membrane? You have to remember, friends, majorly with the help of hydrophobic interactions, which is possessed by the hydrophobic amino acids. With the help of hydrophobic amino acid, they can easily transpan the membrane. And friends, all of you please listen. The C terminal, see, this is extracellular, also called N terminal transmembrane. Cytosolic domain, you can also call it as C-terminal domain or carboxy terminal domain. Friends, all of you please keep it in your mind. This C-terminal domain or the carboxy terminal domain always associated with the G protein. Always associated with what protein? G protein. And friends, if you look into the structure of a G protein, all of you understand G protein is a heterotrimeric. Don't confuse it is a homotrimeric. No. Friends, G protein is a heterotrimeric protein. Hetero, different. It contains 3 different subunits. What are those? Alpha, beta, gamma. All of you kindly look into the diagram carefully. If you see the structure, alpha and gamma always associated with the membrane like this. Always associated with the membrane like this with the help of lipid anchors. With the help of what, friends? With the help of the lipid chains or the lipid anchors. And friends, among alpha, beta, gamma, you can understand the concept very clearly. This particular alpha chain having GDP or GTP binding site. GDP or GTP binding site. Now you have to understand, whenever there is a ligand is present, all of you listen the statement. Whenever there is a ligand is present, then only GPCR get activated. Then only G protein get activated, then only effector get activated, then only there is a production of secondary messenger. So my friends, the unique statement I want you people to remember here is that whenever there is a no ligand, all of you look into the picture carefully here, whenever there is a no ligand, GPCR not activated, G protein is not at all activated because G protein is not associated with the C-terminal domain of the GPCR. But if you look into the another picture, when ligand comes and attaches to the GPCR, immediately there is an activation of the G protein. Friends, immediately what is going to happen to this particular G protein, the G protein alpha, which is in the inactive form. Why I am calling G protein alpha is in the inactive form? Because it is associated with the GDP. You all has to remember, friends, if the G protein alpha wants to convert it from GDP form to the GTP form, it requires an important protein. What is that protein, friends? All of you keep it in your mind. We call it as GEF. What is that? GEF. Guanine exchange factor is the protein which will convert the G protein alpha from GDP to the GTP. Then immediately what is happening, friends? Once the G protein alpha convert from GDP to the GTP form, immediately what's happening? You can understand the picture. The G protein alpha separate from its friends beta and gamma. It completely gets separated. Now the G protein alpha in the GTP form, all of you remember, it will come and activate the effector molecule. So here, friends, in any signaling which is associated or mediated by the GPCR, ultimate goal of the G protein is, friends, to activate effector molecule. That's ultimate goal, to activate the effector molecule. Do not forget the statement. Now the point I want you people to remember here is that, listen to me carefully. Now this particular effector molecule, all of you listen to me carefully. What is the ultimate goal of the effector molecule? Every one of you remember, friends, the ultimate goal of an effector molecule is activation of the secondary messenger. Activation of what, friends? Secondary messenger. Activation of secondary messenger is the ultimate goal of the effector molecule, because you know that secondary messengers are the one which are involved in the signal transduction, and finally they will diffuse into the nucleus and they will show the response of their particular gene. Anyway, that we will understand later. But remember, ultimate goal of the G protein is what, friends? Activating the effector molecule and ultimate goal of the effector molecule is activating the secondary messenger. Friends, this is one of the most important slide where we can expect the questions here. What are the type of ligands that are using different type of receptor, and what kind of effectors are associated with it, and most importantly friends, what kind of a physiological response that it can show. All of you listen to me very carefully friends. First one, epinephrine. You know, right, what is the function of epinephrine friends? Breaking down of the glycogen to the glucose. That's the ultimate goal, we all know very well. So all of you listen to me friends. Epinephrine will show their action with the help of GPCR, but the name of the GPCR is different. That's the only change. What is the name of the GPCR to which epinephrine will come and attaches? Friends, it's a GPCR only, don't confuse. These are all GPCRs only, but they are having a specific names, that's all. This epinephrine will come and associated with a receptor. What do you call it, that particular receptor friends? Beta adrenergic receptors. What do you call friends? Yes Rohit, yes, of course it is useful for the first year, no doubt. You can watch the recorded also. If you in case if you miss Rohit, you can start from the you can start watching from the starting. It's highly useful for the BSc first year also. Epinephrine, it can be used by what friends? Epinephrine can uses the beta adrenergic receptor. Now beta adrenergic receptor is what type of GPCR? As we discussed that now the GPCR is going to get activate the G protein. Now friends, the G protein obviously going to activate a effector molecule. Friends, this is the point every one of you has to remember. This effector molecule only we called adenylyl cyclase. Adenylyl cyclase, ultimately what they are going to do friends, they are going to activate a certain secondary messenger. I am just saying in terms of this. Here the secondary messenger is what friends? Cyclic GMP molecule. Now that cyclic GMP friends involved in the breakdown of glycogen into glucose. That's the ultimate function. Suppose example, you can take serotonin. Friends, serotonin is, you know, right, one of the most important neurotransmitter which can perform major functions of the, which is involved in major functions of the central nervous system. Now the point is very clear friends. This particular serotonin associated with the serotonin receptor, and ultimately serotonin receptor also activating on what kind of effector molecule, adenylyl cyclase. But the response is not same. Response is behavioral sensitization. Now you take another example, light, which is a ligand or a stimulus. The type of GPCR is rhodopsin, and effector molecule is not at all adenylyl cyclase. The effector molecule is cyclic GMP, and the function is what? Visual excitation. And you take a very best example, acetylcholine. It is associated with what? Muscarinic receptor. It's a type of GPCR. Effector molecule, its ultimate goal is what? Activating the potassium channel in order to reduce the pacemaker activity. So what is the statement you ought to understand by this slide? You have to understand a point that depending on the ligand or depending on the primary messenger, anything is same. Depending on the ligand or primary messenger, everything is going to be changed. Receptor will be different, effector molecule will be different, secondary messengers involved is different. Most importantly friends, the physiological response that is showing entirely different. So what's the statement you ought to remember? Depending on the ligand, all the components are going to be changed. Friends, this is the slide where you have to remember the most important examples. Let's talk about the neurotransmitters. It's very difficult to remember everything friends, so I am marking some. I am requesting all of you to remember whichever I marked. Acetylcholine, dopamine, noradrenaline, GABA, 5-HT, MSH, opioids, somatostatin. I am requesting all of you to remember this particular 8 examples without fail. These are all the powerful neurotransmitters which are using GPCR as their major receptors. And If you look into the hormones, what is the point you have to remember? Adrenaline, ACTH, glucagon. Remember once again, adrenaline, ACTH, glucagon, histamine, melatonin and prostaglandin followed by vasopressin. Here I given the examples of 7, here I have given the examples of 8. I am requesting whichever I marked. These are all the most important neurotransmitter and hormones in our body starts using the GPCR. So friends, this is the introduction about the cell signaling as well as the about GPCR pathway, right? Okay friends, that's all for now. We will meet in the next class. So friends, whomever taking the subscription, please kindly use my code Chandu Live, and most importantly, please attend this particular 13th February paper discussion and analysis class without fail. And please kindly take this subscription as limited offer is going on for 12 months and 24 months. Okay friends, thank you all of you. Thank you. I'll see you in the next class. Thank you so much.